Okay, good afternoon all. Stuart Collinson from Oxstem. We are a company focused on innate regeneration and repair. The company was spun out from Oxford University 2016. I joined about 9 months ago, and we are using a small molecule approach to modulate stem or progenitor cells. And what you can see in this diagram here is that as the committed progenitor cell, we intervene around that stage to drive the progenitor to one of the downstream lineages. It depends on the therapeutic area, and I'll get into some of the areas we're working in a little bit later. But the overarching concept is that what we are trying to do is to awaken the body's endogenous, in vivo, innate, call it what you will, but the body's got a certain ability to regenerate. Now, we're not a starfish or a mammal that can actually regenerate a whole limb, a lizard, but the body in certain organ types, tissue types, does have an inherent ability to regenerate. What we're trying to do here is to augment and drive that ability of the body itself to help to cure and treat disease. So what we do is we have a number of therapy areas. The company was originally founded by a triumvirate of chemists, and they then collaborated with experts in specific disease areas. And the choice of the disease area was around not just the medical need, but around the world-class talent existing within the Oxford ecosystem that could become part of this company. So typically we have two founders in each area, one at the translational biology level, the other typically perhaps more at the clinical level. And in each case what we're doing is we're developing very rich content cellular screens. We then screen small molecules against these screens on initially a target agnostic approach. So in other words, what we're trying to do is to look very quickly for a phenotypic effect using these very sophisticated screens, and then we use a small molecule to go back in and try to deconvolute the underlying molecular pathways. I'll talk briefly about three areas: heart failure, diabetes, and neurodegeneration. We have a small team, a small management team of people with a lot of experience across large and small companies, and then we have a large academic body of collaborators that we're working with across these different diseases. So heart failure, a couple of approaches here that we are engaged in. The first is to drive the epicardial cell, you can see on the right-hand side of these images, to a mesenchymal cell. So what we're trying to do here through this so-called EMT transition is to restore lost tissue following myocardial infarction. Now again, the body does have a certain, albeit woefully inadequate, ability to regenerate, so we're using a small molecule to go in and to tickle those progenitor cells, to drive it to mesenchymal cells to rebuild the heart following an insult. That's the first approach. The second approach is lymphangiogenesis. So again, you can see on the right-hand side an example of the quiescent state, and then we go to an activated state following some kind of intervention. So this is a different approach. It's one that could work alongside the EMT transition, or it could be done separately. And again, the idea here is to treat the patient around or post myocardial infarction, but to reduce the tissue damaged by enhancing the inflammatory response resolution, and to just improve the environment of the myocardium. Diabetes, same kind of concept, using a high content screen with a world leader in this area. He spent most of his career working in this area, and the high, the sort of 30,000 foot vision here is patients today, as everybody knows, are trying to manage their insulin. They're using increasingly sophisticated, lower cost, smaller monitoring systems, but in the end it's an insulin injection. And so what we're trying to say, is there a way here where you would not need to monitor and to treat with insulin administration? And so what we're trying to do is to say, look, the beta cell in the pancreas is an exquisite detecting system. It doesn't do just insulin, it does fatty acids, it does nutrients and other molecules. And so what we're saying is, if we could repopulate the pancreas with beta cells, the patient can then go back to a state where they are able to manage their own insulin innately. And so again, it's another regeneration repair story in which we're using a small molecule to drive this pancreatic progenitor cell to a functional beta cell, to repopulate the pancreas with beta cells. And again, as mentioned previously, what we're trying to do here is to use these high content screens, small molecule approach. Once we've got the translational biology sorted out, then we typically move outside the university into collaborators on the CRO side to do more of the high throughput screening. This is early. We are still at the verge of going from the translational biology into a high throughput screen, but making great progress. Neurodegeneration, same concept again. Here we're looking into the dentate gyrus, and we're looking to try to drive cells to create more neurons. The concept here being is that if we can repopulate the neuronal population, then we have a chance to address not all neurodegenerative symptoms. In our case, we are looking at cognition. And this is something that affects a number of neurodegenerative diseases. We've done some initial work in Alzheimer's, but that's unlikely to be our main indication. But going on to the types of screens that we're using, again, you see here the dentate gyrus, the neural stem cell, small molecule intervention going to a functional neuron. And there you can see some pictures on the other side of looking at some of the microscopy and the imaging that we use. In this particular case, we do have a molecule designated here that has now been linked back to a target that is potentially being activated by this approach. So we are starting to get to grips a little bit on the target and the pathways in the neuro program. This is some of the data, and I think this is a demonstration of what the company's been able to achieve so far and what could potentially be applied to other areas. So, start off with in vitro, we go to in vivo, functional correlation. That's the same approach in each area. And this is what we've done here. The protocol is down here, but if you look to the bar charts on the right-hand side, what you see in the top right are basically more neurons in the rodent at sacrifice, and then down below, what you see is a statistically significant correlation in behavior. This is a Y maze test. The rodent goes to the correct arm of the maze as part of this testing. So again, I think good demonstration that we are able to go into the in vitro assay, to in vivo, to the animal models, and in this case as well, start to uncover some of the molecular biology. So that is the three programs. I'm not going to spend any more time. We do have two other programs that we are including here in the appendix. I'll just touch on these very briefly before wrapping this up. Acute myeloid leukemia, you may say, well, what's that got? It's the same kind of idea, cell differentiation. Here we work on the myoblast. We drive the myoblast out of its block status to create the downstream lineages and more of a natural hematopoiesis, so basophils, neutrophils, and so forth. In this case, we've uncovered the target. We don't think it's a target that we think is a good one to prosecute for further work, so we're revisiting the AML program. In the retinitis, this is retinitis pigmentosa. Again, this is very early stage, but very exciting. This is reproducing photoreceptors to try to cure blindness. That concludes the overview of the company. Be very happy to take any questions.